Good afternoon to everyone. This class we are going to study about biodegradable polymers and films and coatings, right? You all know very well, in the recent years, usage of uh, biodegradable polymers is gaining more importance among the consumers because of the uh, hazardous, or we can say that uh, danger of the biopolymer as a landfill. So once the biopolymers are disposed and if as a landfill, what happened to the polymer material? The polymer material enters into the soil and that will affect the aeration of the soil. So plant growth is affected. And when the grazing animals, when they consume them, it chokes the stomach, or it leads to death of the animal is another one. And another thing is, it is also a greatest dangerous to the ocean, water-based animals also. So in order to avoid all these things, all the hazards in all of the forms of our life, we have to go for biodegradable polymer material. So this biodegradable polymer material will degrade in the soil uh, very soon. So it's not going to create any environmental impact to the any of the living sources on the in earth. Okay, so with this background, we are going to study about the biodegradable polymers, what are all the methods involved in the production of biodegradable polymers. All these things we are going to cover in this class. So first, before going into this, as I said, now we have to study about the impact of plastic waste. So you all know very well. The first one is pollution. So plastic waste can pollute the land, air, and water. Okay, so this is the greatest menace nowadays. Okay, so the plastic waste creates pollution in the environment in which it's being disposed. And the second one is harm to wildlife. Plastic waste can harm wildlife in many ways. Animals can become entangled in plastic waste, which can lead to injury or sometimes to death also. Climatic changes. Plastic production and dispose contribute to climatic change. The production of plastic requires fossil fuels. From the fossil fuels, we are extracting olefins. Olefins are used for the manufacture of the polymeric packaging materials. And during this manufacturing process itself, it releases lots of greenhouse gases into the atmosphere. These gases may have greater impact in our the life cycle. That's the ozone depletion takes place. And not only that, the global warming is another reason, because of the emission of the greenhouse gases. All happens because of the plastic waste. Hence, the disposal of plastic waste in landfills is so in the disposal is into the land is a greater menace. And not only that, when during a long time burial into the soil, this actually releases what also produces methane gas, which is another dangerous one. Okay, so and this is the effect, these are all the effects on the environment, and what effects it's going to create on human health. So exposure to harmful chemicals. Plastic waste contains harmful chemicals. This includes bisphenol A and phthalates. So bisphenol A is normally present in any of the water bottles which we are using. So this is a carcinogenic agent. When we consume beyond a limit, it becomes carcinogenic, and it induces cancer in the human body. And also phthalates also like that. Okay, so these harmful chemicals should be avoided. So these chemicals are present in the plastic bottles nowadays. Nowadays, what type of bottles for baby feeding? They are going for a bottles which are all what bisphenol A free, BPA free, like it has been mentioned in the bottle itself. And nowadays they have started manufacturing the bottles with BPA free bottles, like we are getting in the market, right? So this is actually what in the previous lecture we studied about the life cycle assessment. So life cycle assessment mainly concentrates on the prevention of the packaging material, and then what improvement over the packaging material. So free making packaging containers free from bisphenol A is also what? It's an innovative thing and also improvement in the existing method only. The next is increased risk of infections. So microplastics can pollute food and water. They can be ingested by humans. Unknowingly, we are consuming microplastic in our foods, in our food system. No, the thing is that when my plastics are thrown into the ocean water, what will happen? These plastics will be consumed by the marine animals, and which in turn we are consuming the marine animals, and these type of microplastics enters through the marine animals into our body. So this is, we are knowingly or unknowingly, we are what, ingesting the microplastics into in our body. So next is respiratory problems, burning of plastic waste. So in the last one, the methods of recycling we studied. One is landfill, recycle assessment we studied. No, one is we are going to either we are going to use them for landfilling. Second one is incineration. Third one is recycling. So what will happen when you incinerate this plastic waste? When we incinerate the plastic waste, that leads to release of harmful pollutants into the air, such as particulate matters and dioxins. When we consume these dioxins, again, it creates some respiratory issues. Then next is biodegradable polymers we are going to concentrate. So in order to avoid the ill effect which is created by the plastic polymer material to human beings and also environment, we have to go for an alternative method of packaging that is called as biodegradable polymer. Okay. So the biodegradable polymers are nothing but what? They are also a class of material that can be broken down naturally by biological process. Simple process, microorganisms can degrade them. So simple biological process or enzymes into a simpler material. So degradation of the complex packaging into a simpler material by microorganisms and enzymes are called as biodegradation. So this releases non-toxic substances like carbon dioxide, water, or biomass into the environment. And this biodegradable polymer is in contrast with the synthetic non-biodegradable plastics that persist in the environment for extended periods. That means even some hundreds of years, thousands of years, we are not able to dispose them, or we are not able to degrade. The polymers are not able to degrade into the soil. Okay, so that is actually with the synthetic polymer material. The biodegradable polymers are often derived from renewable resources like plants or microorganisms, in contrast to the synthetic polymers. Synthetic polymers are derived from the fossil fuels, petroleum waste. We are extracting them. We are manufacturing olefins, and these olefins are used for the manufacture of synthetic polymer material. But biodegradable polymers are often derived from the renewable energy sources like plants or microorganisms. So they are very safe. And they offer a potential solution to the environmental problem of plastic pollution, as they can easily decomposable. Then instead of creating landfilling issues or something, and pollution problem. So we can avoid, when you go for biodegradable packaging material, you can avoid the environmental pollution as well as also the hazardous ill effect to human beings also. So next we are going to study about the classification of biodegradable packaging material. So bio-based polymers are classified into synthetic biodegradable polymers, natural polymers extracted from biomass, and polymers produced by microorganisms. So biodegradable, bio-based polymers are divided into three categories: synthetic biodegradable, natural biopolymers, then another one is the polymer produced by microorganisms. So what are all the synthetic biodegradable polymers? They are from biomass, either from biomass or from petrochemical things. So the one which is obtained from the biomass is called as polylactic acid. PLA is the bio-based polymer which is obtained from the biomass sources. The next one is the synthetic biodegradable polymers from the petrochemical waste. That is nothing but what? PCl, polycaprolactam, polyvinyl alcohol, and then glycolic acid, or these petrochemical based polymer materials. The next one is called as the natural biopolymers extracted from biomass, or polysaccharides, lipid, Proteins. So the polysaccharide based is starch, cellulose, alginate, carrageenan, and chitosan. And these are all polysaccharide material which are all used for the development of the biodegradable packaging material. And lipid based packaging material includes glycerides and waxes. And based on this, you can make a packaging material. Next is protein. So protein is nothing but gelatin, casein, whey protein, and then soy protein, zein, and wheat, gluten, or some of the protein substances which are all used for the manufacture of biodegradable packaging materials. The final one is the polymers which is being extracted from the produced from microbial are called as microbial polymers, and they includes microbial bacterial cellulose. Bacterial cellulose is nothing but what it is. You will get when you make bacterial cellulose, it undergoes fermentation process, and you'll get a mat of what bacterial cellulose material that can be used as a packaging material. And then polyhydraulics, hydroxyalkanoates, PHA, polyhydroxybutyrate, PHB, and poly-3-hydroxybutyrate and co-3-hydroxyvalerate are some of the microbial synthesized polymer materials. So with this background, we are going to study about how these are all going to be manufactured or manufacturing methods of the packaging material we are going to study. So this is a pictorial representation of the packaging material from different sources. One is from biomass, and another one is from microbial, and another one is from the GNA related things. Sources of biomaterials. So before going to study about the manufacturing aspects, where we are going to get these biomaterials. So biopolymers can be of plant-based, animal-based, and plant and animal-based also. So plant-based, nothing but what? Flour of grains and legumes, fruits and vegetable residues, gums, pectin, starch, cellulose, proteins, sources from plants, or the plant-based biodegradable materials. Then animal-based materials includes marine algae, chitosan and chitin, collagen, gelatin, polylactic, xanthan, gelatin, milk protein, or some of the animal-based biopolymers which are used for biodegradable polymer making process. Then plant and animal-based materials are nothing but lipid materials that can be used for synthesis of biodegradable polymers. So here the table gives about the different types of biomaterials used for the manufacturing of biodegradable polymer material, their sources, and what property it imparts when you make a polymer, and where we are going to use that polymer material are given in this table. Next is fabrication of composite films and coatings. So first one is composite films. The main objective of using edible films and coatings are to prevent deterioration of the fresh produce. You know that when you are coating them, when you are making edible films or coatings, you will naturally cover the fresh produce. Normally, we have to prevent the spoilage of the fresh produce from the environmental agents. Okay, so what will happen when you keep an apple or any tomatoes or green leaves outside the environment? What will happen? Due to the temperature variation, wilting or then removal of moisture may take place from the loss of moisture may take place from the substances. Once you cover them with a proper packaging material, the shelf life can be enhanced. That is the ultimate aim. So the difference between the application of edible films and coatings on food is that films are formed separately by different methods, and then these films are applied on the substances, whereas coatings are directly applied on the materials itself. So the simple or mixed usage of diverse carbohydrates, proteins, or lipid components in various forms like coatings, single layer, bilayer, multi-layer films of coating has been proposed for the manufacture of the edible films and coatings with the regulated barrier properties and also acceptable in high moisture foods, acceptable for high moisture foods also. So first one is film fabrication. First in this class, we are going to study about the film fabrication method, and in the next class, we study about the coating method, different types of coating methods. So one is called as film fabrication method. First one is called as what? So there are different methods are available. One is called as conservation method, simple conservation technique. The second one is called as complex conservation technique, and then gelation or thermal coagulation method, and solvent casting method. Extrusion methods are the methods which are all used for the manufacture of films. Okay, so biodegradable film manufacturing methods or fabrication methods are simple co-observation, complex co-observation. Then you have extrusion method is there, casting method is there. All these methods are mainly employed for the film manufacturing purpose. So first we are going to study about the film fabrication method. Simple co-observation method is the first one. Simple co-observation method is nothing but a micro encapsulation technique. When here a single layer of polymer or surfactant is dissolved in a solvent and then it undergoes liquid-liquid phase separation. And in this, when the phase is getting separated, it forms a denser polymer-rich phase that is nothing but the co-observate and a dilute phase. Okay, so this once a denser polymer-rich phase is developed, the film can be cast from this. This is achieved by altering the condition of the salt concentration, temperature, or by adding a non-solvent. Okay, so what you're going to do in this method, we are going to add a surfactant to the solvent, then it undergoes phase separation into a denser polymer phase and dilute phase. Okay, so this phase separation is achieved by means of what? Altering the conditions like salt concentration, temperature, and adjusting the non-solvent material. So in the simple co-observation process, a single polymer is involved and co-observates are formed due to dehydration or water deficient mechanism. Okay, so in this water deficient mechanism, which is caused by the addition of a salt or desolvation liquid, that is nothing but the co-observation agent or inducing agent into the reaction medium. You have a solvent. To the solvent, you are going to add a surfactant. That surfactant will lead to what? It is going to create what? It's going to form a co-observate, okay, due to depletion of water. Water deficient is created in this case, and then it is going to or else sometimes what happens when you add something to the polymer solvent material, what will happen? Either a salt or a dissolvation. Dissolvation is nothing but what? Solvent separating agent or called co-observating agent or inducing agent. When you add them, what happens? It creates an environment which leads to the dehydration or water deficit of the material which you are using in this case. So here the pictorial representation, see the pictorial representation of this. First process, you are using your polymer solution you are taking. The polymer solution may be of what? Any polysaccharide you can take, any protein base, or any material you can choose. Biodegradable polymer material you are going to choose first. So afterwards, after taking this biodegradable polymer, you are going to heat them, and this periodically what happens? So you have to once it is heated, you are going to add some salt, because we have to desolvent them, or that we have to use some inducing agent or co-observation forming agent. We have to add to this. So next, first you are heating it, then you add some co-observates. Here they are adding salt to it, salt of fatty acids, and then they are stirring it. Okay, once it is stirred, what happens? We are going to add drug solution with the stirred solution. So after stirring them, add the drug solution. Once the drug is added, we are going to add co-observate solution and then stir continuously. What will happen? Solid lipid nanoparticles will be formed in this case. So this is one type of process which is used to form encapsulation of the polymer material, is called as a material. Solid lipid nanoparticles are formed during this co-observation formation, co-observation technique, simple co-observation technique. The second method is nothing but what? You are taking a polymer material, we are going to heat the polymer material. So hot aqueous phase you are getting. So drug containing hot melted lipid phase you are taking. So here we are taking the drug which contains lipid phase is added to this. We are heating them, and then once heated, it is heated, we'll get hot aqueous phase, we'll get it. So we have to stir them, and with the addition of melted lipids, then we have to add lipid to this. First you are taking the polymer material, heat them, then add the lipid material to this. This is melted lipid, we are adding them, and then again we are going to stir them, After stirring, heat the material and then cool them. When you cool this repeated heating and cooling process will lead to the formation of what? So what solid lipid nanoparticles it will form in this case. So this is drug distillation in hot water. This method, product 2 they have given, see, drug distillation into the hot lipid, formation of emulsion, we are forming emulsion. Once the stable emulsions are formed, then pre-emulsion we are forming, heating it and pre-emulsion we are forming, then formation of colloidal emulsion we are making. Once these colloids are formed, then alternate heating and thawing leads to the solid lipid nanoparticles are formed. That means the drug will be coated with the nanoparticles now. So that is going to happen here. If you want to coat any food substances with the polymer material, you can go for this type of simple conservation technique. Here it is a micro encapsulation technique. Here they have given an example of the drug they have given. So drug is added to the material, and once it is added, we are stirring it, emulsion is formed, and then what? Again, we are heating it to form a pre-emulsion, then formation of colloidal emulsion. This alternate heating and cooling results in what? It will form a co-acervates. So the drug will be covered with some what? Some encapsulation will take place in the drug material. The next is complex conservation. So in the complex conservation, we are using when two hydrochloride solutions with opposite electron charges are combined, the polymer complex interacts and precipitate in this case. So that means we are using two hydrochloride solutions. Two different solutions we are going to use, and both the solutions have different charges. So here core disposed in aqueous solution of anionic polymer. So the core material is dispersed in the solution. The solution is anionic polymer material. Okay. Then to this, we are going to add aqueous solution of cationic polymer. We are going to add it. So now we are getting two polymer material, two hydrochloride solutions we are getting in this case with oppositely charged polymer material. Initially, you are taking anionic polymer solution. To the anionic polymer solution, you are going to add what? A cationic polymer you are going to add. So that means what you are doing, you are using two hydrochlorides you are using. Then we are making them to interact between, we are allowing them to interact between these two them. So between two polyions are interacting, and then next what you are going to do, we are going to alter the condition. Alteration of condition is taking place by changing the pH, temperature, or addition of salt. So anything you can do in this case. Once the condition is altered, you will get what? Hardening or cross-linking polymer, you'll get it. That means the polymer material which are cationic, when you are adding cationic, the cationic will be attracted towards the anionic polymer material, and they'll make a complete coating of those things. So the hardening and cross-linking is done next then. So for hardening and cross-linking, we are adding some cross-linking agent. Why we are adding cross-linking agent? This cross-linking agent will keep both the hydrochlorides intact. Okay, so that is the job of the cross-linking agent, so that your polymer will be in a entangled form. Otherwise, they will separate out. For this, cross-linking agents are added. So when you add the cross-linking agent, again co-acervate with the core in the center, a complex co-acervate will be formed. Once you dry it, you will get the dried product. Okay, so this is called as complex conservation. We are choosing two polymer solution, positive and anionic, and also cationic solution we are taking in this case, and then we are allowing them to interact. Once interaction takes place, what happens? Anion and cation will join together, and then what happens? We have to change the condition. By changing the condition is nothing but altering the pH, salt, or temperature. Then add cross-linking agent that will provide a good polymer dry powder of the complex conservate material. You will get it in this case. So the complex conservation is a microencapsulation technique again, where the oppositely charged polyelectrolytes, as I told you already, interact with the aqueous solution, leading to a phase separation and the formation of a polymer-rich shell around a core material. So complex conservation is based on what? The interaction of two oppositely charged polyelectrolytes. The disposed core material is added to the polymer over a narrow pH range. and then mixture is suspended in an aqueous phase containing a surface active agent. So this is first you are taking a emulsion, preparing emulsion when you are altering the pH. So this is the formation of the, this picture shows the formation of the co-acervates, then reticulation by formol. That means we are adding the cross-linking agents, and once this is done, you will get a network. Okay, so this is the final co-acervation microencapsulated product. You will get it. Gelation or thermal coagulation. So in the method, this is the third method in the this process. This is a process in which your macromolecule is heated, and then what? So when you are heating protein substances, what will happen? Denaturation of protein will take place, and then gel formation occurs in this case. So the gelation of proteins, for example, when you heat egg white, what will happen? It creates a gel-like mass. Similar thing is going to happen here. Then, or else when you add something to it, precipitation of the protein takes place, or even the cooling of the hydrochloride dispersion causing gelation in this case. So here the aqueous solution of albumin, when it is heated, and then what? Heat-induced conformational changes, gelation occurs, and protein-protein interaction takes place, and you'll get the albumin nanoparticles you'll get at the end in this case. Okay, so this is the one which depicts the process here. The next one is solvent casting method. So in the solvent casting method, the casting process is a popular method, and also it's an inexpensive method which is used for the making of films. So to make a biofilm, the process entails three phases. One is what? We have to solubilize the form biopolymer into the solvent, and then in a suitable solvent, then you have to pour the solution into a mold, and afterwards what you have to do? You have to dry them. Finally, you'll get a casted film. You will get it. This is the simplest method and low cost method for the manufacturing of the films. Okay, so the steps are described here. First one is solubilization of biopolymer in a suitable solvent. And for example, here you can choose any polymer material, selected polymer material, and ethanol used as ethanol may be used for this purpose. For example, soy protein. If you take soy protein isolate, ethanol can be used to solubilize them. Then afterwards, you are going to heating. Simultaneous heating has to be takes place, and you'll get a gel-like mass. Then you have to what? Cast them into a mold and then dry them. You'll get a film. The next method is called as extrusion method. The extrusion process is based on the polymer thermoplastic characteristics. In general, what happens in extrusion method? We are applying high temperature and high pressure. That leads to what? The making the material into a molten mass. This molten mass, when it comes out of the extruder, it takes a definite shape in this case. So the extruder has three parts. One is feeding zone, kneading zone, and then the last one is called as the heating zone. The advantage of extrusion method is that it is a high performance, low cost, and efficient process which is employed in the food industry for commercial manufacturing process. The feeding zone, the mixture of material is fed into the feeding zone, and it is compressed with air. Then the biopolymer and additives are added to the extruder in this case. In the kneading zone, materials are adequately combined with the help of an extruder screw. Then in the heating zone, the final material is mixed. In this case, the oven is used to supply some heat, so it is finally heated to a molten mass, and this molten mass comes out of the die through the fixed extruder end, deforming the shape and thickness of the extruder film. Whatever may be the die you have, through this it comes out as a material. Okay, so these are all the different types of methods which are all used for making of polymer packaging films or biodegradable films manufacturing technique. Okay, with this we'll stop the class.